So I'd like to start by asking how many of you have heard of a theta state. So if this concept sounds familiar, please put your hand up. And that looks like almost everybody. And so if you've heard that floating induces a theta state, please keep your hand up. And that also looks like almost everybody. So I have some news that might surprise you a little bit. I'm a neuroscientist, but I myself don't know what a theta state is. And that's because theta state isn't a term that neuroscientists use. We do talk about something called theta, but we don't talk about a theta state. So to find out what a theta state is supposed to be, I went online and I consulted this wonderful resource of information known as urbandictionary.com. And one of the first definitions I came across was by My Father's Princess, which says, the brainwave of being in a dream state where you can receive messages from other dimensions. So this is a little bit of a Joe Rogan type definition, and as you can imagine, I was not completely satisfied with this. So next I decided to ask artificial intelligence. And as you may know, there are now many AI programs that will take a text prompt and convert it into an image. So I used the AI program Midjourney, and I gave it the prompt, what is the theta state of consciousness? And it returned these images for me, which, as you can see, completely explain everything. So I'm fully enlightened now, I get it, I know what a theta state is. But getting slightly more serious, let's take a neuroscience perspective on this. So theta is a wave, or more technically a neural oscillation, that occurs at 4 to 8 cycles per second. And it's important to emphasize that theta is not a state, meaning it's not exclusive to other neural oscillations. And this next point might surprise you, but there's actually to date no reliable evidence linking floating to theta activity. Now, you all raised your hands and said you had heard that theta is involved in floating. So where does this association come from? Well, for one, there was this 1993 published report by Tom Fine and colleagues, which is one possible origin for this. So this study ostensibly showed theta and alpha activity in a sample of 9 volunteers during floating, but there were some big problems with the study. So for one, it was not peer-reviewed. It appeared as a published book chapter, but not in a peer-reviewed scientific journal. Secondly, there was no statistical analysis in this study. There was no computerized measure of theta power. And again, this was a small sample of just 9 volunteers. So all of these factors mean that it's possible the authors might have succumbed to confirmation bias and seen what they were looking for. Right? Now, to be fair to Tom Fine, my understanding is that he's since acknowledged the limitations of this study, and he probably never intended for it to start this term theta state. Now, whatever the origins of this theta state hypothesis are, there's unfortunately a lot of misinformation about it online. So just last month, I accessed the Wikipedia article for sensory deprivation. Okay, and you can see in the highlighted text here, there's a statement that doesn't have any reference. So it says, in the last 20 minutes of the float, often end with a transition from beta or alpha brain waves to theta. Now again, there's no study cited here, okay? So there's no reference for this. And unfortunately, in this case, Wikipedia just happens to be wrong. Now, to be fair, there are other reasons why we probably associate theta with floating. And one of those reasons is that there is evidence that theta is linked to inner-directed attention, for example, in the context of meditation. And because many of us, myself included, choose to meditate in the float tank, I think what's happened is we've made the logical jump from theta is associated with meditation, which we have some evidence for, to theta is associated with floating, which we don't have evidence for. So now that I've probably ruffled your feathers a little bit and challenged some of your ideas about theta and floating, I want to give you an outline of what you can expect to get out of this talk. Okay, so first we're going to talk about what happens to the brain during floating. So we're going to look at actual EEG activity recorded in the float tank. And then we're going to test this theta hypothesis. We're going to see if theta activity is actually enhanced by floating. And then finally, we're going to look at whether brain activity recorded in the tank predicts the subjective effects of floating. So at this point, I should introduce myself. Most of you probably don't recognize me. This is my first float conference. So my name is Joel Frohlich. I'm an academic neuroscientist at the University of Tübingen in Germany. And I received my PhD in neuroscience from UCLA in 2018. So shortly after getting my PhD, I did a postdoc in the Monti lab at UCLA, studying consciousness, so specifically disorders of consciousness and sleep. But I also developed this side interest in altered states of consciousness, which is how I got interested in floating. And I'm no longer in LA. I'm now a postdoc at the Institute for Neuromodulation and Neurotechnology at Tübingen. But I am still a consultant for the Institute for Advanced Consciousness Studies in Santa Monica. And let me tell you, those folks are definitely interested in floating. So I'm relatively new to the world of floating compared to most of you. I had my first float experience in March 2020, and this was less than 2 weeks before California issued its first stay-at-home order, so really just on the eve of the COVID-19 pandemic and lockdowns. So my first experience was a 2-hour float at Float Lab in Westwood, Los Angeles. So shout out to Crash at Float Lab if he's listening to this. And as I say, this was a 2-hour experience, and it was my first float. So I had some doubt that I would make it through the whole session. But sure enough, the time just flew by. And as soon as I got out of the tank, I recognized what a powerful experience this had been for me and how it had reduced my own anxiety and induced states of mindfulness and meditation. Now, if you'd like to hear more about my first-person experiences with floating, I've written this article online titled My Out-of-Body Experience. That's in Nautilus magazine, so you can check that out if you're interested. Getting back to the story, so as I say, this happened right at the beginning of the COVID-19 lockdowns. So I had many months of staying at home where I couldn't go float again, but I could reflect back on this experience I'd had, and I could ask myself how I could use my skills as a neuroscientist to better understand floating. So naturally, one of the first things I did was I looked up scientific papers on floating, and I found the work of Justin Feinstein. So many of you know Justin. Many of you know that he's a very friendly guy. So I emailed him. I started collaborating with him over Zoom. And last year, he shared with me these Bluetooth EEG data recorded in the tank that I'll be presenting to you today. So just to dive into some details of this experiment, this happened at LIBOR in Tulsa, Oklahoma. And there were 2 groups of participants recruited. So there were healthy control volunteers, and there were also volunteers with clinical anxiety. And the volunteers completed 2 experimental conditions. So there's a float condition, okay, and the float condition is interoceptive. Now, you may know that LIBOR has this open fiberglass float pool with no enclosure, which is really great for participants with clinical anxiety, okay? It cuts down on the risk of claustrophobia. And there's also work coming out of LIBOR several years ago showing that floating is an interoceptive state, so it enhances internal bodily sensations such as breathing. And this was compared to an exteroceptive film condition in which participants watched this BBC Planet Earth nature documentary. So naturally, if you're watching a film, your attention is directed outward, yeah? So it's an exteroceptive condition. So to go into some technical details, this was a randomized controlled trial, meaning that participants completed both conditions but in randomized order. And we collected EEG data in both conditions. And in fact, during the float, we had usable EEG data from 36 subjects, including 21 subjects with anxiety disorders and 15 non-anxious volunteers. So I'll give you a breakdown of what these sessions looked like. So they're both 90 minutes, the float and the film. And in the float, we have 10 minutes at the beginning where there's lights and music that gently fade out to ease the participant into this stimulus-free environment. Yeah? So that's at the beginning. And then at the end of the float, we have 10 minutes of lights and music that gently fade back in to ease the participant into a normal sensory environment. So it's these middle 70 minutes here that are dark and silent. And so that's where we chose to analyze the EEG data from. Now, for those of you who have floated, you know there's 3 stages, roughly speaking, to this experience. There's this pre-float baseline, where you feel relatively normal, presumably. Then you have what happens during the float itself, which, if you have a good session, includes some interesting psychological effects. And then you have this, then you have this post-float afterglow, if my slides will go back to it, where you have these after effects of floating. Okay, and the after effects of floating have been studied already by Justin's team at LIBOR using fMRI neuroimaging. But what's really difficult is to study what happens during the float itself, okay? So to illustrate how this is non-trivial, I want to use a more intuitive example. So you might ask yourself if you've ever wondered whether the light in your refrigerator stays on when the door is closed. How would you ever know? You could open the door and look inside, but then you're ruining the very closed environment that you're trying to study. So with floating, we face a similar challenge. We can look inside the tank, open it up, put some instruments inside, but then we're probably going to ruin the very stimulus-free environment that we want to study. So I want to talk now about two common technologies for studying brain activity in the laboratory. One is fMRI, or functional magnetic resonance imaging. Okay, this is commonly known as brain scanning. So fMRI has very good spatial resolution, and it produces these pretty pictures of the brain that you see online and in the news, and it looks at what parts of the brain are using the most oxygen. Okay, but unfortunately, fMRI has poor time resolution. It's rather like watching a movie that's just 1 frame per second, and we cannot use it to study these neural oscillations, such as theta, which are 4 to 8 cycles per second. And furthermore, if you've seen an MRI machine, you know that it's this enormous magnet that weighs as much as an elephant, right? And the local magnetic field produced by the MRI machine is so powerful that it has to be kept in a specially shielded room away from metallic objects. So obviously, we're not going to deploy MRI in the float tank, okay? We can have subjects get out of the float tank and then go into the MRI scanner. Okay, that's a study that Justin's team at LIBOR has done before, but we cannot use MRI to study what's happening during the float itself. So an alternative technology is EEG, or electroencephalography. So EEG is lightweight, it's portable, and it's inexpensive. Okay? And it has low spatial resolution, meaning it doesn't produce these pretty pictures of the brain, but it has very high time resolution, meaning we can precisely look at the timing of neural events, and we can study neural oscillations, such as theta. Okay, so far so good. But there are a couple challenges to deploying EEG in a float tank. So one is tactile sensation. Here you can see this picture of me wearing an EEG cap. And this cap holds the electrodes together that are pressed against the scalp to record electrical brain activity. And you know, it's quite tight. There's usually a chin strap that holds this in place. And furthermore, there's this conductive gel that has to be pipetted underneath each electrode. And all of this creates a lot of tactile sensation, okay, which kind of defeats the whole point of going in a float tank in the first place. So the next challenge is called bridging. All of you know that the water in the tank is highly saline, and these salts can act as an electrolyte, okay, so they can carry current between electrodes. And so if we have EEG submerged underwater in the tank, these electrodes are going to become electrically coupled. Okay, so this means they're all picking up the same signal. It would be rather like you're watching TV, you're flipping through the channels, and every channel is showing the same TV program. Okay, so obviously that's not a desirable thing. Now, fortunately, the company Neuroverse has come up with a very elegant solution to this problem in the form of a product called the Brain Station, which you see right here. Oh, whoops. Yeah, right here. So you may know Neuroverse from Ricardo, the CEO, who's spoken at previous float conferences. And the Neuroverse product, Brain Station, it's attached to the forehead, it stays above the level of the water, and it's completely wireless, okay? It uses Bluetooth technology to record EEG signals. So the Brain Station solves the problems that I've outlined already, okay? So there's minimal tactile sensation, there's no cap, okay? Nothing like that, it's just this little device that fits on the forehead and records frontal EEG. There's nothing submerged underwater. We don't have to worry about bridging, and it's only one channel. Nonetheless, it's designed to be waterproof, and it's designed to be salt-proof. So it's wrapped in tegaderm to protect it from salt. And it's wireless, so this is Bluetooth technology. There's no wires, and there's no cables. All right? So now that I've told you how we can use EEG to record signals in the float tank, I just want to give you a brief primer on how to think about EEG like a neuroscientist. So you probably already know, I'm just going to make this more intuitive, you already know that you can take white light, send it through a prism, and you get these different colors or frequencies of visible light out the other side. And in the same manner, we can take the EEG signal, send it through this mathematical tool called the Fourier transform, and we get these different EEG frequencies out the other side. So I'm just going to go through the different EEG frequencies. The lowest frequency that's commonly studied is called delta. Okay, so this is 1 to 4 cycles per second, and it's often associated with states of deep unconsciousness, like dreamless sleep and general anesthesia. But there are some big exceptions to this, which will become important later in the talk. So I want everybody to understand that no one frequency band is associated with just one function. Okay, that's important. Next, there's theta, which we've already talked about, but just to recap, it's 4 to 8 cycles per second, and it's often associated with states of inner-directed attention, such as meditation. After theta comes alpha at 8 to 13 cycles per second. Alpha is associated with neural inhibition. So for instance, if you close your eyes, the visual parts of the brain become inhibited, and you see these very high amplitude alpha waves over visual cortex. After alpha comes beta. Beta is 13 to 30 cycles per second. It's involved in various functions, but most prominently bodily movement and the motor system. And then finally, we have gamma, which is 30 cycles per second and upward. So gamma is involved in cognitive functions like attention and working memory, but it also overlaps with the frequency range of muscle activity. And a lot of times, EEG is also picking up muscle activity in addition to brain activity, which means that in EEG studies, we need to be careful to see that the gamma activity is really coming from the brain. Okay, so I know I've hit you with a lot of information about neural oscillations, but I still want to give you one more primer on how to understand the EEG figures that I'll be showing you today. So these are called time-frequency representations, and they're tools that show the EEG power at different points in time and in frequency. So they look like this, okay, rectangular plot, and on the horizontal axis of this plot, we have time in minutes, yeah? And then on the vertical axis, we have frequency in cycles per second, or hertz. And notice that when we go up in frequency, we go up logarithmically, which means rather than counting linearly, 1, 2, 3, 4, 5, we count logarithmically, 1, 2, 4, 8, 16, 32. And the reason we do that is because it results in more even spacing between the different EEG frequency bands. Now finally, there's this color bar here, which shows the strength of the EEG signal at different points in time and frequency. And this is often set up as a contrast between two conditions. So in this example, we have a contrast between float and film. It's float minus film. And so where we see warm colors here, like these reds, oranges, and yellows, yeah, this is where we have more EEG power in the first condition, the float condition. Where we have these cool colors, these blues and cyans, this means that we have less EEG power in the first condition, the float condition. And where you see green, you have approximately the same power between these two conditions. Okay, so just to recap, I've overlaid the different EEG frequency bands on top of this time frequency representation. So we have the lowest frequency, delta, down here, and we have the highest frequency, gamma, up here. And because we have this on a logarithmic scale, frequency is logarithmic here, we have roughly even spacing between the different EEG frequency bands. So now if I take this overlap away, what we're looking at right now is the contrast between the float condition and the film condition in the healthy participants in this study. So I know this is a rather technical figure, but just study it carefully, see what patterns you can glean from it. I think the first thing you'll probably notice is that there's a lot of warm colors here toward the bottom of the figure. And remember, when we're looking at the bottom of the figure, we're looking at these low frequencies, okay? So that means that floating enhances low-frequency EEG activity compared to the film condition. And then if we look at the top of this figure, we see that there's a lot of these cool colors, these blues and cyans. And that means that in the high frequencies, like gamma and beta, we see attenuation of EEG activity in the float condition compared to the film condition. Yeah? And you might ask yourself what frequency you think this enhancement is happening in at the bottom, okay? And you might be thinking about theta, but if you look at the vertical axis, you can see a lot of these warm colors are between 2 or 1 and 4 hertz, okay? And if you recall, that's actually in the delta band. Not the theta band, but the delta band. So now, what I'd like to show you is the contrast, the same contrast between float and film, but in the participants with clinical anxiety. And I think you're going to see the same pattern even more clearly. So again, these are really technical figures. So I'm going to give you a moment. I'm not going to talk. Just really study this figure, try to see what patterns you can glean from it, and take a moment to digest it. Okay, so I'm sure all of you probably see that there's a sea of red at the bottom of the figure, right? So even more clearly than in the healthy participants, you see that there's this low frequency enhancement of EEG activity in the float condition compared to the film condition. So the same pattern as we saw before, but even more clearly. And just like what we saw in the healthy participants, there's this high frequency attenuation in the float condition compared to the film condition. Now let's go back to this low frequency enhancement. Again, what frequency do you think this is happening in? I'm sure a lot of you are coming at this with the theta hypothesis. So you might be guessing this is happening in theta, but if I overlap the theta band on top of this time frequency representation, you see that theta actually misses almost all of this red, okay? It's missing most of this low frequency enhancement. So remember, everything below theta is in delta. So what we're actually seeing is delta enhancement with floating in these participants with clinical anxiety. Now, as scientists, we like to do statistical analyses to see that our results are unlikely to be random occurrences due to chance, yeah? This can get rather technical, but I'm going to spare you the nitty-gritty details. And what you need to know right now is this is a time-frequency representation where we now have red outlines, these red contours surrounding clusters where you see significantly more power in the film condition compared to the float condition in both groups of participants. So if you take a look, you can see that these are rather broad clusters both in time and in frequency. So they span almost the entire duration of the session. And if we look at the frequencies they encompass, there's gamma, beta, so these high frequencies that we would expect to see. But then you also see a little bit of alpha, and you even see a little bit of theta. Now remember, these are the frequencies that are being enhanced by the film condition, not the float condition. And now if we look at the clusters that are significantly enhanced by floating relative to film, we're now concentrating on, again, these red contours here. This is at the bottom of the figure, okay? So there are several of these clusters, and they're quite low frequency. And again, you might ask yourself what frequency you think this is taking place in. And I'm sure many of you still want to think that this is happening in theta, but if I put the theta band on top of this figure, you see that it misses almost all of this activity. Okay? So these clusters are almost entirely beneath the theta band. So this is almost entirely taking place in the delta band, where we have significantly more EEG activity during the float as compared to the film. Okay, another thing we can look at is something called group by condition interaction, which is simply to say that we see slightly different effects of floating in one group versus another. And you can see this visually for yourself. I have the healthy plot on top of the anxiety plot. So in the participants in the control condition, you see that there's a little bit less of this low frequency enhancement than what you see in the participants of clinical anxiety. You can just tell that qualitatively from your eye. But where this really gets interesting is when we look at frequencies where there's significant attenuation of EEG activity in the anxiety group. okay? So that's these new clusters you're looking at here, surrounded in red. You can see that there's a cluster at the beginning of the float, okay, which encompasses some high frequencies and some lower frequencies, and then about 50 minutes in, you have a couple more clusters. And these clusters seem to be centered on the alpha band, okay? But you also have a little bit of the theta band here. And remember, we're looking at the frequencies that are attenuated by floating. Not enhanced by floating, but attenuated. So you're actually seeing that floating doesn't enhance theta activity. If anything, it's attenuating it. So I'm sure at this point, many of you are a little bit confused. You're scratching your heads and you're asking what's going on. And you might be saying, how is it that an EEG marker of inner-directed attention and meditation is actually being attenuated rather than enhanced by floating? Well, the first thing I would say in response to this is that meditation and floating are not necessarily the same thing. Many of us choose to practice meditation in the float tank, myself included, but floating is a broader activity. Nonetheless, let's talk about meditation for a moment. So there was this great review of the literature in 2015 by Lomas and colleagues, and they looked at different studies that examined EEG theta power in meditation. And as you can imagine, they found quite a few studies. It was 10 or 11 studies that reported increases in theta power during meditation, okay? However, they also found a handful of studies that reported no difference between theta and a resting condition, or between meditation and resting condition with regard to theta power, okay? And even more surprisingly, you even find a few studies that report decreases in theta during meditation. So what's going on here? Well, there's a lot of heterogeneity that needs to be understood. So for one thing, it matters quite a bit whether we're looking at participants who are novice meditators, complete beginners, right? Or if we're looking at experienced Buddhist monks. And we should expect those two groups to have different neural signatures. And it also matters quite a bit what meditation tradition we're studying. So as I'm sure you know, there are many different styles of meditation, and it might make a difference whether we're looking at Vipassana or Metta. Okay? So if I can now take your attention away from theta and back to delta, I told you at the beginning of this presentation that we would see whether EEG activity recorded in the float tank predicts the subjective effects of floating. And that's what we're going to see here in this figure. So on the horizontal axis, we have delta power, okay? And on the vertical axis, we have the change in anxiety. So the change in anxiety was measured using the Spielberger State Anxiety Inventory, which, by the way, was the primary outcome measure of this study when it was pre-registered on clinicaltrials.gov. And just to help you understand this vertical axis, so where you see 0 here, this is no change in anxiety. That means the participants felt the same getting out of the tank as they did going in. No change in anxiety. Between 0 and 10, this red area, we have increases in anxiety. That means these participants felt worse getting out of the tank than they did going in. And between 0 and negative 40, we have decreases in anxiety. And that means the participants felt better coming out of the tank than they did going in. Well, the first thing I'd like you to notice is that all of these magenta points are participants with clinical anxiety, okay? And if you look at these, they all had decreases in anxiety, okay? None of them felt worse getting out of the tank than they did going in. They all felt better. The next thing I'd like you to notice is that if we look at the correlation between delta power and the change in anxiety, it's a negative correlation. So what that means is the more delta power you had during your float, the bigger a decrease in anxiety you experienced. Now, I would want to see this replicated in a larger sample before I really commit to saying that, yes, the delta power definitely predicts the change in anxiety. But this is still important because it suggests that the delta power, rather than just being incidental to the effects of floating, it may really be something that's involved in the anxiolytic effect. So for the nerds in the audience, this was a Pearson correlation of negative 0.3. And just to show you that this is specific to floating, we can look at the same relationship, delta power versus anxiety change in the film condition, and there's no significant correlation. Okay, so this is really something that's specific to floating. All right, I know I've hit you with a lot of information, and This is probably a little bit disorienting, understandably so. So I want to just pause for a moment and emphasize a few take-home points. So first of all, I hope it's obvious now, but floating induces delta activity. Not theta activity, but delta activity. And there's actually no reliable evidence to call floating a theta state. So I think for this reason, we should stop calling floating a theta state. And furthermore, I think we might want to start thinking about floating as a unique state of consciousness and not necessarily put it in the same box as meditation. Now, I'm sure many of you are probably still a little bit hung up on this delta finding. And you might be thinking to yourself, wait a minute, delta, isn't this just about states of unconsciousness and dreamless sleep? Well, no, it is true that you see delta activity during unconsciousness, but there are many exceptions to this, which I mentioned earlier in the talk. So before I had any interest in floating, I actually did this research looking at delta activity during conscious states. And a couple colleagues and I at UCLA, we did a review of the literature, and we published this review in the journal Brain. It's called Consciousness Among Delta Waves, a Paradox. It's behind a paywall, but I'm happy to email it to anyone who's interested. And we found many different published studies that report EEG delta activity during wakeful consciousness. And you might be very interested to know that one condition in which we observed EEG, or in which the literature reported EEG delta activity during consciousness, is the psychedelic drug dimethyltryptamine, or DMT. So now that we have Joe Rogan's attention again, let's talk about DMT for a moment. So DMT is a psychedelic like LSD and psilocybin, and induces this rich visual experience along with perceived encounters with other entities. But unlike some psychedelics, when DMT is smoked, it induces this sensory disconnect between the mind and the environment. So this is a Dutch woman who just smoked DMT on YouTube, and you can see 3.5 minutes after smoking DMT, it looks like she's sleeping, but she's actually not. So she's having this very rich internal visual experience. And there's some very interesting work coming out of Imperial College London by Chris Timmerman and colleagues, showing that when you give DMT to volunteers in the lab, they have this increase in EEG delta activity. And furthermore, the strength of this delta activity positively correlates with the intensity of the DMT trip. Now, the difference between this delta activity that you see in DMT and the delta activity that you see during states of deep sleep is that in DMT, you see this delta activity that's low amplitude. So we would say it's less than 100 microvolts. And during deep sleep, you see this delta activity that's high amplitude, okay? It's often called slow waves, more than 100 microvolts in amplitude. Now, as it so happens, there's a sensory disconnect between the mind and the environment that all of us experience every night, okay? It's called REM sleep, or rapid eye movement sleep, okay? This is the stage of sleep where you're the most likely to be dreaming. Now, I've already told you that we see delta wave activity during non-REM sleep, when we're not dreaming. But it turns out we also can see delta waves in REM sleep. This was not appreciated until recently, but in 2019, there was a study by Bernardi and colleagues in the Journal of Neuroscience, and they described delta waves during REM sleep. And again, this is a lower amplitude delta wave than what you see in non-REM sleep, or dreamless sleep, okay? So in REM sleep, this is, again, less than 100 microvolts. And what I think might be going on here is that this low amplitude delta activity is a neural signature of an altered state of consciousness in which there's a sensory disconnect between the mind and the environment. And of course, when we go in a float tank, we also have a sensory disconnect between the mind and the environment. That's the very point of going in a float tank. And we also sometimes have this internally generated imagery, which I'll talk about in a few slides. Okay? So I think this might be an explanation for what's happening in the float tank, yeah, with regard to delta activity. Nonetheless, a hard skeptic could still look at these data and say, how do we know these participants weren't just sleeping? Couldn't it be that everybody was taking a nap? And this is a question that I want to take very seriously, okay? It's a reasonable question, especially because from my own experience, I can often be in the tank and feel like I'm in this very ambiguous state of consciousness. I'm neither completely awake nor completely asleep. So the study dealt with this in a few different ways. So for one, all the participants were explicitly instructed not to sleep. But, of course, that doesn't mean that they didn't. So they were then asked afterwards to estimate the proportion of the float that they spent sleeping. And the average response was that they slept for 24% of the session. So on average, they're awake 3/4 of the float, and they're sleeping 1/4 of the float. But the good news is that the EEG delta activity does not correlate with the estimated sleep duration. Okay, so you can see this here, delta activity on the horizontal axis, and then estimated sleep duration on the vertical axis. There's no significant correlation, and in fact, if we try to fit a line through these points, if anything, we see a negative slope. The more delta power you have, the less sleep you have, but it's not a significant correlation. For the nerds in the audience, this is a Pearson correlation of negative 0.1. Another point which I think is relevant is that these same participants with anxiety disorders, in a previous study of floating, they were interviewed afterwards about their experiences in the tank. And many of them had conscious experiences of light in the tank, which we know is completely dark. Okay, so one person said, I saw little flashes of light like starbursts randomly in different places. It was beautiful. Okay, so this person is having a visual experience that they're recalling in the tank. And while I'm not suggesting that this is dreaming in the conventional sense of the word, it is possible that some of these participants are being pushed to this razor's edge between wakefulness and sleep, known as hypnagogia, which is often characterized by these sorts of mild hallucinations. So a future direction for further studies would be to actually investigate whether floating induces hypnagogia. Okay, I know it's been a long talk. I've hit you with a lot of information. Probably some of it is disorienting. And I think at the beginning of this talk, we started with a very opaque and blurry image of what's actually happening in the tank. And I'm hoping that in the course of this talk, I've really clarified what's happening in the tank with respect to EEG signals. But there are a couple more technologies that I would still like to see deployed in the tank. One is called FNIRS, or Functional Near-Infrared Spectroscopy. So FNIRS is looking at optical absorption of infrared light by the brain. And I'm optimistic that we might be able to deploy this one day in the float tank. Another technology is called OPMs, or optically pumped magnetometers. So these are extremely sensitive magnetic instruments that can detect very, very weak biomagnetic fields. And in theory, we could put them in the bottom of the float tank, maybe a couple centimeters beneath the person's head, and we could actually use them to record neural activity. And this would be the best case scenario because there would be nothing touching the person's body as we're recording this neural activity. And then finally, I think we should also use EEG to study the after effects of floating. So remember, there's this three stages of floating, right? We just looked at what happens during the float, and Justin's team has used fMRI to look at the after effects, but I would also like to use EEG to look at these after effects of floating. Okay, so long talk, a lot of information here. I know many of you are probably feeling a bit disoriented and questioning what you thought you knew about floating and theta. But I just want to emphasize some conclusions here. So first of all, I think I've made this clear, but floating is actually not about theta, okay? It induces delta activity, but there's no evidence that it induces theta activity. Now, delta might relate to the anxiolysis that we see with floating, okay? We saw this with this correlation between EEG delta power and the change in anxiety. But there's no evidence right now linking it to sleep, okay? We do not see a correlation between delta power and estimated sleep duration. So I've said this before, and I'll say it again. I think we should stop talking about floating as a theta state because we don't see evidence for it, right? And I think we might want to think about floating really as a unique state of consciousness. We don't need to put it in the same box as meditation, and we don't need to put it in the same box as sleep. And finally, by the way, if I ask artificial intelligence to draw the float state of consciousness, it also looks beautiful. Thank you very much.